So, I have this picture of Alex, and, it's almost the same pose that you had in that picture in the movie. I like this picture for two reasons. Number one is it sort of brings home, I think, to folks how challenging this is to tackle. When I first saw Alex, of course, this is nothing compared to Kim's feelings about this, but when I first met Alex, it was pretty overwhelming how many challenges he was facing and how many things we had in front of us that we had, how many problems we had to solve and work on and make progress on. And also, the second reason why I like this is I think Alex was, with that finger of his, making a point, right? And which is, you guys need to get going on this. Okay, so as Kim mentioned, we started on this 20 years ago between our group and the foundation. And when you're faced with a very complex problem with a lot of things going on at the same time, it can, as I mentioned, be overwhelming. And so what we tend to do is try to break it down into parts and components and divide it up and try to tackle things one thing at a time. And these are on a kind of a course level, some of the problems we faced back then, and I bet any of you parents could make this list 5 to 10 times as long, right, of challenges that you're up against. But this is kind of what we thought our job was to do these things. So the first one we set out to tackle is what is Proteus Syndrome, which is now kind of hard to believe, but in 1997, we didn't know what Proteus Syndrome was. And the crazy thing is that most people, and initially us included, didn't know that we didn't know what Proteus Syndrome was. And that was a rather shocking realization from that first large clinic that we had. And we saw a large number of patients. And we began to put together this notion that, my goodness, what everyone thinks Proteus Syndrome is more than one thing. And to understand that and study it, it is absolutely essential in biomedical research that you're working on one problem. Because if you're working on multiple problems simultaneously and you don't know it, it makes that problem much harder to solve. So I just put up as some representative examples here some feet. The feet are one of the easier things that we use as a sign of this disorder. Kids with Proteus on the left and kids with overgrowth disorders, not Proteus, on the right. And when we started, all of them were thought to have Proteus Syndrome. And we, along with our dear colleague Mike Cohen, began to figure out at that first meeting what was and what wasn't Proteus, although I will admit that the moms in the waiting room figured it out before we did. Right? When they saw the kids coming and going, they were whispering to each other, that kid looks like my kid, or that kid doesn't look like my kid, that kid looks like the other kid. And they figured it out. So nothing magical about it, but it was absolutely essential that we bring these people together at one time in one place, look at them carefully, and then we could see that. So that led to this concept here, which is a little bit tedious and dry, but was absolutely essential and really helped the field enormously and our colleagues. And Kim, you mentioned that people in Colorado Springs can define what Proteus is. Well, in 1997, almost no physicians and almost no genetics doctors could define what Proteus Syndrome was. So, I mean, that's how far this has come. So we've proposed these, which turned out to be incredibly useful for us as researchers and clinicians to work toward figuring out the cause of the disorder. But on a practical level, it also helped enormously because it turned out that kids with true Proteus syndrome and kids with other overgrowth syndromes that look like Proteus syndrome are medically pretty different and need to be managed differently, and it helped us to do that better. It also then coupled into this notion, which was not invented by us, but by dermatologist Rudy Hopla, that this disorder was mosaic and that our understanding of the biology of this disease could be based on this theory, which we then set out to work on. Okay, so we got one thing off the list. Now, with all of these things, I'm going to sort of X them off as I go, but I do want you to understand that none of these stories are completely done. All right? We are still working on all of these, including the first one that I just X'd off. We are working on a couple patients that have, of course, as patients always do, we doctors and scientists set out these wonderful rules for how things are. And as soon as we set a nice rule, a patient comes along and breaks that rule. That's their job, right? So there are some exceptions to this, and we're working on that, and you'll hear about that eventually. But that problem is mostly solved. So once we were clearer on who had Proteus syndrome and who didn't, then we set out to work hard on figuring out what problems these patients have and how those problems evolve over time, because those things are critical for us to understand in order to take care of patients. You have to know what problems they're likely to have and how they change over time in order to predict what's going to happen and therefore to have a better idea of what to do. One of the big ones, and one that we tackled pretty effectively, is this. This is for those of you who aren't radiologists, this is a CAT scan, and this is a rather terrifying CAT scan. Physicians who see this image kind of shake in their boots because what this is a CAT scan of a patient's chest. This is not from a patient with Proteus syndrome. I just got it off the internet. But this is a patient who has a pulmonary embolism. All right, and what you're supposed to see, the blood vessels are these white pipe-like things there, and what you're supposed to see is solid white, and what you see in that white pipe is gray globs. Those gray globs are blood clots, and that is a very, very serious complication, and that's what led to Alex's demise. And so we recognize through difficult experiences that this is a huge problem, and got very aggressive about treating that and have been able to manage that to the extent that I think this is much less of a problem than it used to be, and in fact was a huge problem previously for any patient who wanted to undergo surgery. This happened not infrequently post-operatively, and now we're at the point where we've done I don't know how many operations on patients, and cross our fingers, knock on wood, we haven't had a significant complication of this since, and that's a huge advance for patients with this disease. So this is an effort that we undertook, supported by Arcule. It was critical for the approval process for the drug to try and understand exactly what the survival is with Proteus syndrome. And we could talk around this, but I wouldn't fool any of you families because we all know this is a very serious disorder. And we have a challenge in front of us that this disorder is shortening some patients' lives, and not in a rare or infrequent way, but in a pretty common way. And we have to be honest about this and alert for this and do everything we can to make this change. So there's lots of examples of this. I scratched this one out. This one deserves probably even less of a scratch than the previous one does. And Kim's going to talk more about some of the other manifestations because we are still learning a lot about the details about what medical problems arise in patients with this disorder. and how to understand those better over time, and you'll see later that that couples into the trial. Okay, third point, the third thing we have to tackle is what can we do for patients to make them better even if we don't know what's causing the disorder. So that's called symptomatic treatment. This is not Dr. Tosi. I don't have a picture of Dr. Tosi in the operating room. I should have, but I don't. Again, thank goodness for the internet. But we, early on, did a number of surgeries on patients with Proteus Syndrome for two reasons. Number one, to benefit those patients symptomatically. And the orthopedics was by far the best example of that, and the rather remarkable procedures that were done that helped patients enormously in improving their mobility and their growth was a huge benefit, and we are working hard to try and get that information out to Dr. Tosi's orthopedic colleagues so that that can get better for everyone. And for sure, we have more work to do there, even though I scratched it out. So what causes Proteus Syndrome? So you have all seen this more than once, and as the folks who visited my office know, and I have this framed on my wall, and Kim has one. I don't know if it's even hung.
It's in my front room.
It's in your front room, okay. And Tracy Whitewood-Neal has one as well. This is the result, incredibly nerdy, geeky picture. For those of you who don't do science, it's kind of pathetic, but we just get so unbelievably excited when we see something like this. These are the sequence data that Kim mentioned in the movie, and this is the actual screenshot of the work that was done primarily by Marge, showing the variant in patient 5. And patient 5, this patient 5, just so we're clear, is not patient 5 of the drug study. Okay, so there's two patient 5s, so be careful with that. And the answer is right here. You can see at the top it's supposed to be a C, and instead there's a mix of C's and T's, which told us the answer to that question. And the answer is another really nerdy picture, this. And our gene that is causing this disorder is, as many of you know, AKT1. And this cartoon diagram is meant to communicate to you. It probably looks like gibberish to most of you, but it is intended to communicate the fact that what AKT1 is a molecule in the cell whose job it is to listen to what other molecules are saying and tell other molecules what to do. And it shouldn't have been the slightest surprise to us, and it made perfect sense when we found it, that the molecules that AKT1 are listening to are growth factors. Okay, these are proteins that circulate in our body that tell the cells when to grow and when to stop growing. And so growth factors circulate around the body. They land on cells. Cells have receptors that receive those growth factors. They understand that a growth factor is there. It's telling the cell to grow or to move or to expand. And that receptor then talks to another molecule, and then that other molecule talks to AKT1, and AKT1 does a whole bunch of stuff to the cell. And the problem is, with Proteus Syndrome, is that AKT1's kind of like, I'm just going to say it, kind of like one of those mother-in-laws. Just never stops talking. And the genetic change in AKT1 causes AKT1 is supposed to be quiet sometimes and talk when there's a growth factor. Instead, even when there's not a growth factor, it's talking all the time. So it's turned on, it doesn't listen, and it just keeps talking. And that is biologically what leads to the problems in Proteus syndrome. So that then explains an enormous number of things about Proteus syndrome, what that change is doing, and importantly, it gives us an idea about what we could do to change that, to treat it. So then we have an understanding of what Proteus syndrome is, and then we have to ask the question, can we treat it? Is there something we can do to make that molecule stop doing what it's not supposed to be doing, or at least do it enough less that it could make things better? So the first thing, and again, I think Margie talked about this 2 years ago, right? Yeah. This was done again primarily by Marge in the lab, which is that cells from patients with Proteus syndrome were taken into the lab and put in dishes and with culture media, and cells kind of look like this. These are the cells that we grow out from patients with the disorder. And then we take those cells and we measure how much AKT1 is talking. And when AKT1 has the Proteus syndrome mutation, here's a little graph, it's talking a lot. If you add a compound to the cells in culture, this ARQ092 that we've been talking about, AKT1 doesn't talk as much. All right? That's a short way of explaining a very long series of experiments that Marge did. So this gave us a hint that we could use this compound on cells from patients with Proteus syndrome, but there's a huge difference between doing this in dishes in the lab versus doing it in a human being. There are a lot of hurdles to get from this to this. So, patients, where do we start? How do we approach that problem? So the first thing we have to figure out is, in a lab, you can just put whatever you want on the cells, and you don't really care how much they complain. You don't care if the cells die. You just try things. You can't do that in the clinic with patients. So you have to figure out where to start, how much to use, what kind of an approach you're going to take. The huge challenge here in Proteus syndrome is adapting from one use of a drug to another. And I talked about this a little bit 2 years ago, but it's really important to review that. What we're trying to do here is to identify a treatment that will work to suppress this disorder. Because now we understand what causes it, now we understand how it changes over time. And what we all think is that this drug is not going to be a drug like an antibiotic, where if you have an infection, I give you a drug, I get rid of the infection, 10 days later, you're off the drug and you're cured. That is extremely unlikely to ever be the case with something like Proteus syndrome. It is also extremely unlike cancer therapy. Cancer therapy is more like an infection. You have a tumor. We're going to treat you, if you have a tumor, with a drug. You're going to be on that treatment for a relatively short period of time. We are going to do everything we can to get rid of that tumor, and then when the tumor is gone, you're off the treatment and you're cured. Okay? Proteus syndrome is a lifelong condition, and our objective will be to control that and treat it, but we don't think we will cure it, but we do think that we do have the potential to stop it in its tracks, which itself would be a pretty awesome and incredible thing. It's also possible that we could potentially reverse some of the manifestations, but it will almost certainly require treatment for years, if not decades. So if we are going to be thinking about developing a treatment for a disorder that is going to require treatment for years or decades, then it becomes something that's more like diabetes than it is like cancer. right? We don't make diabetes go away. We give patients a treatment that keeps them healthy and keeps diabetes from killing them, because it can, and they're on that treatment for their entire lives. And that treatment is incredibly successful and has added decades and decades of healthy lifespan to those people, and that's going to be our goal here. So if we're going to ask people to do that, just like in diabetes and not like in cancer treatment, the treatment has to be very tolerable, has to have very low side effects, and has to be easy to administer and accept. We can, and I'm going to be too crude here, pardon me if I'm wrong, Brian, but in those of you who've had relatives who've had cancer probably understand this all too well, which is the crude way of thinking about what cancer therapy is we poison patients to within an inch of their lives to try and get rid of their cancer. And the hope is that the poison is more poisonous to the cancer than it is to the patient. And then you kill that cancer, and then it's gone. That's worth doing because it is such a dreadful disease. It's worth that side effect. No way on earth can we do anything like that to patients with Proteus syndrome. So we have to start at a completely different place and think very strategically about how we can do this in a way that will be effective and very, very tolerable, make it more like diabetes treatment than it is like cancer treatment. So we have this very optimistic and aggressive approach we're trying to take to safety and tolerance, is that we want to have very, very low side effects and a very tolerable treatment. So we started at a dose that was about 1/12. Is that the correct number? Yes. 1/12 that for this drug is being used in patients with cancer is where we started in Proteus syndrome. And that's a really small fraction. In drug terms, that's like a world of difference away. And the reason for that is that it was based on the experiments that were done in the lab by Marge on the cells, as well as the pre-human treatment data that the company had that showed where this drug goes, how much of it stays in the blood, how much of it goes in the tissues, primarily in animal models. And we put all those things together and we said, we think we should start out at about 1/12 the dose that the cancer patients are getting. So then we have to say, okay, that's what we want from a safety standpoint for a starting dose. Then we need to say, how are we going to measure a successful outcome in this very first trial? So we set these things that are called primary and secondary endpoints. So a primary endpoint is the main objective of the trial, such that if that thing works, you call the trial a success. If it doesn't work, the trial is a failure. So our primary endpoint was somewhat like what Marge did with the cells in the lab, which is that what we wanted to do was ask the question of, can we give the patients this low dose of this drug to the extent that what we can actually do is go into the patient and measure how much it's turning down the AKT1, how much that talkative protein is talking less, okay? We also set out a number of goals, which we call secondary endpoints. These are things that we can do and that we will try, and it doesn't matter if they work or not so long as we learn from them. These are called secondary endpoints. So these are like bonuses, right? Bonus questions on your test. If you get them right, you get extra credit. If you don't get them right, no penalty. So what we actually said as secondary endpoints is actual treatment outcomes. And the main goal there in this first trial was to try and understand and test some ideas about what we could measure in patients, because what we have to do in the long run is show with a measurable outcome, not just we think you're better or you think they're better, but we actually measure something that is a problem in Proteus syndrome, and that it's a measured level at one point, and then after treatment, it is not as bad, improved or better, or stable if in real life it normally progresses. So those are our secondary endpoints. Okay, so how did it work? So safety. This was phenomenally successful. The safety of this drug in these patients at this dose level was better than I could have possibly imagined. And I was thinking about this, that, well, these patients have very few significant side effects. Everybody has symptoms when they're on a medication, whether the drug is causing them or not, because we all have symptoms of things. And patients with Proteus syndrome have symptoms more than healthy people do. So we saw things in patients with Proteus syndrome, but when we actually measured them, they were very surprisingly low. And I was thinking last night, comparing this to the other trial that Dr. Kepler's running, where we're using a drug that's already approved and is in use clinically with patients with overgrowth disorders and other treatments, a very widely accepted drug. ARQ-092 at this dose in Proteus syndrome has much less in the way of serious side effects than does this other approved drug that we're using and that's already in the market. So I think that's a pretty stunning comparison to think that this experimental drug, and we were anxious and worried about if things would go wrong and if patients would have side effects and it would cause problems, but truth be told, it caused less than another approved drug in an overgrowth syndrome that people generally accept. So that's a huge advance. Primary endpoint, making AKT1 not talk so much. Okay, this is a graph of a result in one of the patients that we have treated, and what it shows is that before treatment, is my arrow on? Oops. Yeah, there it is. So before treatment, you can see the drug is up here. I mean, the AKT1 is up here. It is talking a lot. Okay? Then after, oh no, I'm sorry. This is day 15. I had it wrong. So this is day 15. This is showing that after 2 weeks of treatment, we didn't see much change. And then after, I can't read that number. What's that number? Thank you. My angle's terrible. That it is half as much. All right? And that was exactly our target, is that we wanted to have in the patients the AKT1 signaling level, the talking, be about half what it was before treatment. And overall, in 5 out of 6 patients at either day 15 or day 90, we were just about exactly at 50%. So that shows that our estimates when we started were really surprisingly accurate, right? Again, using the extensive animal data that we had, using Marge's cell culture data and drug level data and things like that, the prediction we made that about 1/12 of the dose is where we should start turned out to be almost exactly right. Now, the trial was designed to assume that we wouldn't have guessed correctly. We picked 1/12 because that was our best guess, but we were not very confident that was the right number. And so the trial was designed, if it wasn't right, what we would see in these results is that the dose was too low and there was not enough effect on the AKT, in which case we would have to do it again with a higher dose, or if the effect was too strong or the patients were having side effects that we thought were too high, then we would have to lower the dose and try again. So we don't have to do, because we actually got it pretty much exactly right with the first dose, which is pretty remarkable. Okay, secondary endpoints. We'll start with the bad news first. Several of them just didn't work. All right, and they didn't work, and that's okay because they're secondary endpoints, and we were setting out to learn how to do this stuff. We had patients go in the MRI scanner, and we were trying to measure the size of their bones, because of course, you know, bone growth is a huge challenge in patients with Proteus Syndrome. And what we were trying to do is see if we could use MRI scans to measure the volume of their bones and see if that was going to either decrease or stabilize in patients with the disease. It turned out to be technically much more difficult than we thought it was. The scan time is long. That's a pain for us and for the patients. And the precision, the accuracy of that measurement just isn't good enough for this purpose. So that was a bust. We also measured quality of life. There's a standardized scale which the patients had to evaluate and rate quality of life. And we haven't done the final analysis yet on that. The first look at it is that it's probably not a precise enough measure of how the disease is changing or not. It's not sufficiently precise to be useful. And we also had patients going to physical medicine to actually measure range of motion in some of the small bones and large bones. And similarly, I'm not convinced, again, I don't think we had the final analysis on that. I'm not thinking that that is going to work for us. So these things failed, and that's okay. We learned from it, and we're actually using these data to already design how we're going to do it better in the next trial. So that's the bad news. So then the good news is here. So the infamous cerebriform connective tissue, Nevis, Dr. Darling invented for us a quantitative technique for measuring those. And we measure two things. Those of you who are familiar with this lesion know that there's the obvious CCTN, where it's that lumpy brain-like looking lesion. But before it does that, it actually has sort of a pre-CCTN abnormality, which looks like this. And what it is that there's like a very subtle lumpiness to the skin, with the visible abnormality being kind of like a pearly tissue underneath the skin that you can kind of see and feel. And you can see that sort of white blotchiness to the skin. All right, so that's the pre-CCTN. So here, this is before treatment, both of these. So here, Dr. Darling can measure the size of the CCTN itself, the obvious CCTN. And this is the whole thing, including that sort of pre-CCTN. So we do both. And what you can actually see, which my jaw just about fell to the floor when I saw these data, is that after treatment, the area that was pre-CCTN was smaller than it was before treatment. And here, on the actual CCTN, I don't know if you can see that very well. The resolution's not super. The actual grooves and the depths and the degree of the bumpiness of the lesion before treatment is worse than it is after. Okay? So it's a little smoother, it's less CCTN-like than before. And then the other thing subjectively is the patients say, and I think you felt, that the lesions actually felt a little softer. Okay? One of the things you know about CCTNs is they're pretty darn firm. And that's because of the incredible density of the cells that are in it and the connective tissue. And so that hardness seems to be reversing. And so again, this is a jaw dropper for us, because what we had kind of hoped, at least with the initial trial, was to find some outcome that would arrest the disease, just stop it. But what we're actually seeing here is some sign, and this was present in two patients, this is the more dramatic of the two, that it actually is reversing to some extent. Okay? And I quizzed my colleagues this morning. I don't think in 20 years we have seen anything in any Proteus syndrome patient get better without treatment in any patient we've ever seen. So this is huge. Okay, so we now have some good reason to think that we can treat something in Proteus syndrome. And so where we think we're going to go now is we think that this has been sufficiently successful that we don't need to mess around with raising or lowering the dose. The dose is pretty much right where it ought to be, and that we have from the biopsies that were done and the AKT1 levels that were done, our primary endpoint, which is that 50% reduction in the signaling, we succeeded in, and we even succeeded in one of the secondary endpoints. And so what we want to do is, as fast as we possibly can, move on to the next stage of trial design. So we need to do several things in this next trial design. The first thing is we need to focus on younger patients. We are incredibly grateful for the older patients who started this. That was absolutely the right thing to do from a patient safety standpoint. It's always better to have adult volunteers and people who can understand risks that they're taking and be those sort of brave pioneers to try something first. And what we learned in this study is that the changes that we're seeing, it's the small numbers and it's hard to be confident about this, but the changes that we're seeing were more dramatic in the younger patients, and that's where we think the real benefit can be over time. And that's what we want to be able to do, is to be able to begin to treat patients as soon as they're diagnosed and prevent this disease from progressing at all. So we're going to shift the age range. We need to now make the clinical outcomes the primary endpoints. Right before was those biopsies and the stuff in the lab. Now what we need to do is make the actual patient outcome be the primary endpoints. CCTN, this method that Dr. Darling invented, is a great tool. We're going to continue to use that. The MRI of the bones, we're going to dump that. And Dr. Kepler has done great work exploring. There's a new CAT scan technique that now is available and wasn't available when we started this trial because that's an X-ray technique. There's limits to how many X-rays we can use when we're doing studies on children especially, but this gets around that. Plus, we're going to have to think creatively about how to measure things in other patients based on their unique Proteus medical problems and try and get customized endpoints for different patients who have other problems. We can start, I think we're pretty good, as I mentioned, with this drug dose as the starting dose, and then measure them over time for stabilization and improvement. And then what we will do is what all of your doctors do with drugs that they treat you with all the time, which is to escalate, increase or decrease dose based on your treatment response. So we'll have these clinical outcome measures. We will measure them, and then after a suitable period of time, if we don't see improvement or we see worsening, we can increase the dose. Or if we see improvement and we see any side effects, we can decrease the dose. That's the same thing your doctors do with every drug they treat you with. So that's where we are. The people who made this trial work, I put the main players on the left in alphabetical order. It's impossible for me to rank them in importance because they were all critical to making this work. We have had quite a number of post-baccalaureate trainees in the lab who've helped with a lot of things, too many to name. A lot of clinicians have shared and sent their patients to us. The foundations and are cool, so thank you all very much. Oh, the other comparison I wanted to say, and Brian, I need a number from you. Overall, in the industry, first trials of drugs on patients, first in disease, what fraction of them show signs of efficacy?
So I was doing that the other day because I've brought about 15 drugs through.
Yeah.
only 4 of them had any kind of efficacy signal that you would say wasn't just by chance or wasn't spurious. So it's relatively low. I think for a disease like this, it's even something that is completely new and novel, it's even lower. So this is amazing. It's really, really exciting for me, and this is what I get up in the morning to try and do every day. Try and find something like this.
So these trials mostly don't work, and the fact that this seems to show us some sign of working in some patients, that is an amazing outcome. Questions? Kim?
So if we get to the point where we can give this drug as soon as the child is diagnosed, do we think that it can prevent even things like the bone growth and even things like, so we think it actually...
There's reason to hope for that, absolutely.